Good afternoon everyone. Now I'm going to tackle about gene regulation and mutation. This is the main idea. Gene expression is regulated by the cell and mutation can affect this expression. Gene regulation is the ability of an organism to control which genes are described in response to the environment. We have a prokaryote gene regulation. Prokaryotes are simple. In prokaryotes, an operon often controls the transcription of genes in response to changes in the environment. An operon is a section of DNA that contains the genes for the proteins needed for a specific metabolic pathway. A regulatory gene turns on and off transcription. Eukaryotic gene regulation. When we say eukaryotic gene regulation, eukaryotes have many more genes than prokaryotes. How is transcription controlled? Transcription factors type of protein ensure that a gene is used of the right time and that proteins are made in the right amount. The complex structure of eukaryotic DNA also regulates transcription. Ayan. This is the image of controlling transcription in eukaryotes. Some transcription factors guide the binding of RNA polymerase to a promoter. This determines which protein will be made. Hox genes or home-box genes. Hox genes code for transcription factors and control differentiation process through which all cells become specialized in shape and function. Hox genes are used in embryo development and are active in different zones of the embryo and control what body part will develop in different parts of the embryo. Hox genes are responsible for the general body pattern of most animals. And we have an RNA interference. When we say RNA interference is another way eukaryotic genes are regulated in RNA inference. Small segments of RNA bind to mRNA and prevent parts of mRNA from being translated. RNA interference can stop the mRNA from translating its message. Recall that every cell contains an entire set of the organism's DNA. Only a small segment of the DNA is used for each specialized cell to do its job. Only the part of the DNA that is needed will be used in protein synthesis. Mutations. A mutation is a permanent change that occurs in a cell's DNA. A mutation in a gene sequence protein synthesis can change the protein that is made. Types of mutation: point mutation, insertion, and deletion. A mutation in DNA changes the amino acid base sequence. A different amino acid series of amino acids are incorporated into the protein that is made on the ribosome. The result can be a malfunction of the protein. Mutated proteins often do work. Remember, the shape is very important to its function or ability to do its job. A mutation can affect a single nucleotide or a large segment of DNA. We have a types of mutation: points mutation. Points mutation based pair substitution, a mutation in which one nucleotide base replaces another. Point mutation affect only one codon, so they affect only one amino acid in a peptide chain. It may or may not have serious effect on an organism. It depends on where the mutation occurs and how it affects the protein for which it codes. It can be harmless. Muscular dystrophy is an example of a disease caused by a point mutation. Muscular dystrophy is progressive muscle disorder characterized by the weakening of many muscles in the body. Types of mutation or insertion. Insertion is the addition of one or more bases into a nucleotide sequence. This can change many amino acids in the polypeptide chain, thus changing the protein. It causes a frame shift in the RNA bases, thus causing the wrong amino acid to be added to the polypeptide chain. Disease is caused by insertion. Is a chronic pain, nausea, fever, and weight loss. Types of mutation or deletion. Deletion, one or more bases is removed from nucleotide sequence. A different amino acid is put into peptide chain, thus changing the protein. Cystic fibrosis are disease characterized by abnormally thick mucus in the lungs. Intestines and pancreas is an example of a mutation caused by a deletion. And we have frame shift. Both insertion and deletion result in a frame shift. A frame shift is simply a shift to the left or right in the nitrogen bases, resulting in new codons. A frame shift changes the number bases in a sequence. We have a types of mutation or chromosomal. Point mutation, insertion, and base deletion affect only a single gene or segment of DNA. A chromosomal mutation affects the entire chromosome. Chromosomal mutation change the number of genes in a chromosome or the organization of the chromosome. We will cover more of this with genetics. So this is the image of major types of mutation. Ayan. The order of amino acids is similar to the order or letters in words in a sentence. If a slight change happens, the meaning can change. So this is the mutation. Yan. Fragile X. An example of a mutation is a Fragile X. Fragile X syndrome is due to many extra repeated CGG units near the end of the X chromosome, making the lower tip of the X chromosome appear fragile. Protein folding instability. Substitution can lead to genetic disorder. Substitution can change both the folding and stability of the protein. An example is sickle cell disease. Compare the order of the amino acids is the normal change versus the mutated chain. What is the difference? Ayan. Causes of mutation. Mutation can occur spontaneously. DNA polymerase can make a mistake. Chemicals and radiation also can damage DNA. High energy forms of radiation such as X-rays and gamma rays are highly mutagenic. We have body cell V and sex cell mutation. Somatic cells mutation are not passed on the next generation. Somatic cells are cells in the body that are not next cells. Mutation that occurs in sex cells are passed on to the organism's offspring and will be present in every cell of the offspring. The mutation may not change how the cell